It's impossible to go from a D to an A star is something that I've heard teachers tell students. However, it's not impossible. It is incredibly hard, but one of the main reasons that students who do make that level of progress is they focus on doing the right kinds of revision and exam technique. And that's why in this video, I've bundled together all of my exam technique videos. So I frequently get comments saying, how should I do application questions, or the comprehension, or the long answer question, or those really tricky evaluate questions? And I've got all of those videos out there, but I don't think people are finding them. So to help you out, all of the exam techniques that you need to know are now in this one video. So you can get settled in, comfy, get your notes, get yourself a tea, and let's get ready to improve your grade. If you do need even more help improving your grade, particularly with the exam technique, I do have a monthly membership, the Biology Study Club, which involves multiple live lessons a week, some with me, some with other extremely experienced teachers and examiners. But for now, let's get improving your exam technique. In this video, I'm going to be going through how to approach application questions. So this is actually an exam technique video rather than biology specific. So if you are new here, click subscribe so you don't miss out on any of the latest videos. So first of all, what we mean by application questions, and it's typically applying your knowledge. And it falls under what we call AO2, or in other words, assessment objective number 2. And these assessment objectives are the same across all the exam boards for AS and A-level biology. So although I specialise in AQA, this is relevant for any exam board. And what AO2, or Assessment Objective 2, means is the ability to apply your knowledge and your understanding of scientific ideas, procedures, techniques, and processes. Now, just to show you, this bit is specific for AQA, but it's going to be similar for all the exam boards. And we've got here the percentage of questions, which will be AO2 on paper 1, 2, and 3, and therefore the overall weighting approximately for your entire A-level. So you can see that almost half of your A-level will be application questions. And this is one of the reasons why students are really requesting one of these application videos. Now, if you do want to jump straight to the modelled example, I've got the time codes at the bottom, so you can just click and jump straight to that. But what I'm going to go through before we look at an example is a technique to use to try and make sure you are much more confident and you're gaining more marks. So the first thing is how you actually prepare for these before an exam. And because the whole point is you're applying your knowledge, you have to have really secure subject knowledge first of all. And that is of all the topics, all the theory, but also all the practical techniques. Because if you have a look, they said AO2 is applying your knowledge of not just the scientific ideas, but techniques and procedures as well. So the way to go about that is making sure you are really going through this active recall consolidation, or in other words, revision, after the lessons and in the lead up to the exams. And I'll link my video up here for my top 7 active recall techniques based on neuroscience. So that's one thing, you do have to have really good subject knowledge. The second thing is you have to have practiced these questions repeatedly. And when you practice them, note down the common key phrases or key words for each topic that you notice coming up, and then repeat those same questions at a later date. So it's all about having really strong knowledge and practice. So that's how to prepare for them. But when you're actually doing the exam question, I do have 4 top tips on how to try and make sure you do get full marks. So the first thing is, when you're reading the question, application questions typically have a bit of information at the start where it's a completely unknown bit of theory. And what you have to do is identify the topic, or it could be the experiment, the practical, that this unknown bit of information links to. So that's number 1, identify which topic it links to. Step number 2 is, for the topic you've identified, you need to recall the key words and phrases which you know are really important for that topic and get you marks. So for that, what you can do then is, when you are revising for each topic, just note down all the keywords. And in all my videos, I do emphasize the keywords if you do need help with that particular aspect of this. Now, number 3 is where you're starting to do the application. So you need to have a look at the question, start to think, how could you apply those keywords to fit this question? And that links to number 4 as well. You need to be able to identify which information from the question to include in your answer as well. Because it's not just going to be you stating facts and keywords linked to the topic that you remember. You have to be able to link it to unknown information that is given to you in the question. So I'll go through an example so this makes a bit more sense. So here is my application question. And we're told that gout is a disease caused by the build-up of uric acid crystals in joints. Uric acid is produced from xanthine in a reaction catalysed by the enzyme xanthine oxidase. And then we're given the chemical reaction. We're then told that there is a drug here that is used to treat gout, and we've got the diagram showing the structures of the xanthine and the allopurinol drug. And the question, this was 3 marks, was use this information to suggest how allopurinol can be used to treat gout. So as soon as you see suggest how, you know it's an application question, because you have to use your knowledge, use the information to suggest an answer. So as I said, the first thing you should do is identify the topic this unknown information links to. Then the keywords that are linked to that, then how you'd use those in the questions, and then which bit of information from the question you're going to use. So if we have a look, first of all, identifying the topic, this is an enzymes question. So we've got there in the information it's to do with enzymes. So that is the first thing, we've identified that. Then we have to go through what are our keywords and phrases that are linked to enzymes. So the keywords and phrases for that are unique shaped active site. The active site is complementary to the substrate shape. Enzyme substrate complexes, now that one is key, there's always a mark for correctly using that phrase in an enzyme question. And then also tertiary structure protein. So these are some of the key phrases that I've picked out that I think might come up in the answer. So the last bit is looking at what is the relevant information from this question that we might have to use, and then we'll look at how we'll use that information and the keywords to formulate an answer. So I've highlighted what I think is the key information. So the word equation and the fact that the diagram shows the structures of those 2 chemicals. So now we use all of that to come up with an answer. So we need to use that information that I've highlighted, use these keywords to suggest how allopurinol can be used to treat gout. So my answer here, what I'm going for is, first of all, xanthine and allopurinol have similar shaped structures. That's how I've used the information. You can see they both have rings within their structures. Next then, what is the relevance of this? So the fact they have similar structures means that the allopurinol will also be complementary to the active site shape, and therefore it's able to bind to the active site. Now this actually links to another topic which I've slotted in. That is a description of a competitive inhibitor. So I'm putting that in as well, another key phrase. Now if we have a competitive inhibitor bound to the active site, that then means we cannot form enzyme substrate complexes. So that's how I'm using that phrase. And if we don't have enzyme substrate complexes, the reaction cannot occur, or now we link it to the word equation, uric acid cannot be produced, and that is how it's treating gout. So that's just one example to show you the stages that I would use, and it's all about your subject knowledge using those key words. So comparing this to the mark scheme, you can see I have included more information than is actually needed, but I would always do that to cover all bases. And the marks were pointing out it's a similar shape to xanthine, pointing out that it can bind to the active site or it's a competitive inhibitor, And then we can see this mark here, there's fewer enzyme substrate complexes. In this video, I'm going to be going through the exam technique critical analysis. So we'll have a look at some ways that you can lay out the questions to try and better address the question to get full marks, as well as some typical types of questions that you get. So the critical analysis exam technique, in particular, is on paper 3, and you'll have at least 15 marks of critical analysis questions. You do get some critical analysis in the other papers as well, but the main focus is in paper 3. So what we mean by this then is when you get these questions, like these two here. So it always starts with some information, normally unknown, but linked to one of the theory topics, and it tells you what the scientists are investigating. It gives you a bit of information about their method, and then it gives you their results. And we can see that over here as well. So the scientists are investigating the effect of caffeine on heart rate in human volunteers. We then have a bit about the method and the results. So that's how the critical analysis questions always start. What's been investigated, method and results. And the types of questions that come up with this could be using the information to suggest reasons why. And that could be using the information in terms of the method to suggest why they used a particular control group or why they used a particular piece of equipment. Or it could be suggesting reasons why for the results, which also links to this point here, explain the results. You often get questions linked to the control experiment as well, which would be when you have an additional group set up who aren't being given, if it's, for example, over here, caffeine, they're not being given the active ingredient, which is caffeine. So you're then just looking at the results to see or to compare the effect of the independent variable. And the final type of question, which actually I've got some other alternatives. We've got use the information to evaluate the conclusion, but that could also be phrased in these other ways as well. So do you agree with the conclusion? What can you conclude from the data? And do you trust the conclusion? So those four are essentially the same question, just phrased differently. And this is the big one that we're going to focus on because you nearly always get asked one of these four linked to a critical analysis question. So the way that I always start introducing this in school is with this mind map. So I lift off the data and the information from the question, and I put it into this mind map where we have the first box is describe, the second box is conclude, and the third is evaluate, which is where you're coming up with how confident you are in your conclusion. And I deliberately do this to get students to think about what elements they need to be looking for when they read the information and when they look at the data. Because when you get these critical analysis questions, that is the first step. You need to be reading through the information with purpose. So by that I mean underlining the key points which you think could link to and evaluate, so a pro or a con with the method. Really focusing on what the scientists are investigating, because that will link to your conclusion. And then the description is for the data, so highlighting or annotating what the key patterns are. So you could do this as a practice with paper 3 AQA questions, with these critical analysis questions, just focusing on picking out the description, conclusion and evaluation. And when you do that, it will be quite slow. It might take you 20 minutes before you even get onto the question. But the whole point is this is an exam technique you have to practice, and it will be slow at the start. But the more you practice it in this way, it will become second nature to you, that when you read the information, you'll know to pick out the description, think of a conclusion, pros and cons. And then when you actually do it in exam conditions without this mind map, you'll be much quicker and you'll start to spot exactly the patterns that you need for your answers. So I'm going to go through this worked example here, but instead of having it as a mind map, I'm actually going to do it this way round, which you might prefer, just having it as sections to pull out the information. So the information we have is scientists investigated whether there was a change in the communities of 3 different habitats between 2019 and 2020. So the conclusion straight away, that's going to link to what they're investigating. So we're going to have to conclude once we've examined the data, whether there was or was not a change in the communities. So conclusions are always very short. Describe, so we'll then have to look at all of this data and pick out what are the key patterns, trends, results. It's basically you're stating the obvious. What are the key patterns? Once we've then picked those out, I'm going to jot down what I think the conclusion is. And then the last thing we do before we even get on to the question is picking out from your descriptions which of those are providing evidence to support the conclusion you've written, and which of those descriptions do not provide supporting evidence for your conclusion. So I'm going to go through what I mean exactly by those three boxes. So first of all, I'm going to pick out my descriptions, and I've highlighted as I've gone. So the first thing is the obvious patterns I can see are that the mean species richness increased in habitats B and C, but it decreased in A. That's what the positive and negative symbol mean. So that's my first description that I can see from the results. The other column though is to do with the p-value, and the p-value tells you the probability that this change is due to chance. So the p-value is something that they would have got after doing a statistic. And in this case, it would have been the student t-test. So what I can then describe from these are that the change in the mean species richness for habitats A and B is significant. And that's because there is less than 5% probability, and in this case, there's less than 1% probability that those two changes are due to chance, and therefore it is a significant change. So that leaves my final description. For habitat C, the mean change in species richness is not significant. And we know that because we have more than 5% probability the difference is due to chance. And if it's over 5%, it is not significant. So from that then, I need to look back at what they're investigating to come up with a very brief conclusion. So I'm thinking here, we've got the scientists investigated whether there was a change in the communities. Now, there was a change that was significant in 2 out of the 3 habitats. So that's what I'm going to conclude at this stage, that there was a significant change in A and B, but not C. And then the last bit to pick apart before we get to the exam question is to then evaluate this. So which of your descriptions support the conclusion? Which of the descriptions do not support the conclusion that there was a change in the communities? So I've colour-coded this. So in red, those are my pieces of information which do not support the conclusion that there was a change in the communities. And in blue are my pieces of evidence that do support the conclusion that there was a change in the habitats. And you can see each time I have used all of that information. I've said whether it's a decrease or an increase in the species richness, and I've actually quoted exactly what the p-value means. So less than 5% probability, it's due to chance, or 1%, and this one's 5% again. So that is what I would be doing when I was practicing critical analysis questions, really taking the time to analyse the information, whether that's the method or the results in detail, and split it into describing the data, coming up with a conclusion, and then evaluate your conclusion based on the data. And as I said, that will probably take you about 20 minutes each time, but that's the whole point. It's practice to improve on this exam technique. Now, the actual exam question linked to this is, the scientists concluded that the communities did change. Using only the data in the table, evaluate this conclusion. And now, because I've already done all of this, I can just lift this information from the evaluate box, and that would be my 3 marks straight away. So that's one of the advantages. It then makes answering the questions a lot quicker because you've already done the legwork at the beginning. It also means you've got a better understanding of the method, what they were trying to find out, and what the results show. So you're more likely to give an accurate answer and a better understood answer, and therefore your clarity would have improved. Today we're going to go through the evaluate exam style questions. So first of all, it's thinking about what do we mean by evaluate in biology, because it does mean different things for different subjects. So here we have the A-level definition from the AQA exam board. So they state that students should use the information supplied within the question, as well as your own knowledge and understanding, to consider evidence for and against when making a judgment. Now for A-level, one difference compared to GCSE is that you don't then get a mark for giving your overall conclusion, which would be the judgment. It's just for providing evidence for and against. These questions are normally worth somewhere between 4 and 6 marks in the exam, and you don't actually have to give equal pieces of evidence for the for and the against. But if it was a 4-mark question, then 3 of those 4 marks would be the maximum you'd be allowed to have on either side, for or against. So it doesn't have to be balanced, but you can't just provide evidence for or just against. And there's 3 key examples that I'm going to be going through, and that is evaluating the data, the method, and the conclusion. And those are the key questions that you'd see. So for evaluate the method, I'm going to go through with you the general points to look out for. And that would be points to do with the sample size, whether there are repeats, the control or the control experiment, control variables, whether there's any bias, and the duration of the experiment. So I've given here a method as an example just to demonstrate how to identify some of those points. So first of all, sample size. We're told here that a team of scientists are investigating the effectiveness of 4 different influenza vaccines. And they had a total of 1,800 people, which was split into 5 groups, so 360 was our sample size. So you might be commenting that 360 isn't actually a massive sample size for a drug trial. So that would be going against in terms of evaluating the method, whether you trust it. The sample size is quite small. The next thing that we can see here is we're told that the 5th group was a control group or the control experiment. So they were given just a saline solution instead of one of the active drugs in the vaccine. So that would be an advantage because we do have a control experiment, and that's useful because it means that any differences that we then see between each of these vaccines is due to the active ingredient rather than the placebo effect. Duration of the experiment, so they have given us information on the duration, and they've said that the next year they're going to see how effective it is when they're exposed to the next year's influenza virus. Now, the things to consider though as well that they haven't mentioned, I'm going to go through. So they haven't said anything about whether they've repeated this year on year. So you could have that as a negative. There's nothing there about the repeats. They also haven't stated anything about the people in each group. We know there's 360 of them, but we don't know, are they male or female? Are they older or younger? And do they have any other previous health conditions which could affect how well the vaccine works for them? So that would be a negative. There's also no mention of whether the investigation was conducted with a blind or double-blind trial, and this is when the blind trial is if the volunteers do not know whether they have the placebo, which is the saline solution in this case, or one of the active vaccine jabs. And a double-blind trial is when neither the volunteers or the doctors, or in this case the scientists, know who's got which, and therefore it avoids bias. So if you are asked to evaluate a method, these are some of the general points to look out for. If they have discussed it, you then need to decide whether they have got a good sample size, for example, have they repeated it enough times, and is it biased or not, and so on. Evaluate the data is more commonly asked than evaluate the method, and I'm just going to go through the general points first. So the beginning mark you will always have to state is the overall trend, and this would be evidence for the conclusion that you have. And in this example I've got, we can see the overall trend is a positive correlation. And in this example, the overall trend is a difference. So we can see that the MV plus MMR group is lower than the MMR only group. So we can see that MMR only has a higher mean amount of antibodies. So when you are evaluating the data, that will always be mark number 1 for stating the overall trend. And I've put down here as a reminder, you should always be using data as evidence for your points. Now, for a positive correlation, you won't necessarily be using data, but you should be using the labels on the 2 axes. So we'd be saying there's a positive correlation between pH and the number of water beetle species. Okay, general point number 2 is there any overlap in the data? And I've highlighted this on the scatter graph so you can see it more clearly, because in this case the answer is yes. There is an overlap in the data because for the number of water beetles at 16, we can see we have multiple data points for the different pHs. And if we have a look here for the same pH, we have a variation in the different number of beetles. And those overlaps go against the positive correlation. Now on this one here, any overlap in the data, we can't actually see this particularly clearly without standard deviation. So for this, any overlap in data, for a bar chart, you'd want standard deviation bars to be able to comment on that. The third general point that you should always be looking out for is comparing your control group to your test group. Now, neither of these 2 examples actually show the control experiment, so I can't demonstrate it here. But what we could look at is looking at the lakes without fish compared to the lakes with fish. And at the lower pHs, there doesn't look like there is any difference in the number of water beetles at those different pHs, because that's actually not showing any particular pattern at all. You might also comment on the fact that the lakes with fish have data points from pH 6 up to 7.5, whereas lakes without fish don't. So the last general thing is, was there a statistic used? And if so, what does it use? Now in these 2 cases, there isn't a statistic. Now even if there is not a statistic, you still mention it, because what we could then say is, there is a positive correlation, however, the statistic, Spearman's rank, or the correlation coefficient, was not conducted, and therefore we do not know if it is a significant positive correlation. Same idea here, we don't have any information as to whether a statistic was used, so that would mean we'd be able to say, although there is a higher mean amount of antibodies, without a statistic, we do not know if that is significantly higher. So those are the general points, but for each question you would have to pick out some specific points to do with the graph or the data in a table that you do have. For example, the one that I said earlier about the fact that we only have data points for the higher pHs for one of the lakes. But you could also say that we only have 5 data points, or actually 6, which isn't really enough to be confident in your positive correlation. So the final option is a combination of the 2 we've already looked at. Evaluate the conclusion means consider both the method that was used to collect the data and the data itself to come to a judgment. So that could be evaluate the conclusion, or they might ask you, do you trust the conclusion? So you need to consider both of those aspects, unless in some cases the question might say evaluate the conclusion using only the data, in which case you'd revert back to evaluate the data. The other thing I want to emphasize is, in these questions, you need to use all the information you have been provided in the question. So all of the written information about the method and all of the data in tables or graphs as well. And sometimes that might be spread across subsections of the questions. For example, you have question 1, part A, part B, part C, part D, and the final bit, evaluate the conclusion, might be part 1E. But some of the answers might be all the way back at the beginning, which could even be on the previous page. So if you're asked to use all the information, make sure you look back across the whole question so that you haven't missed any points. This video is going to be going through graph questions, and it will include exam question walkthroughs as well. So many students dread these graph questions. They might have really good knowledge, but when they get to a graph question, they just get stuck. So that's why I'm doing this video, to give you some help and just try and give you some tips so you can get full marks on these questions. So the most common types of questions that you get linked to graphs are either describe the graph, use the graph to explain something, use the graph to calculate, or using the data from the graph to conclude. So I'm going to go through a range of these examples. Now your time is precious while you are revising, so I've put time codes at the bottom so you can jump to any of these in particular that you think you might need more help with. Now first of all, an overview of what we actually mean by these command words. So describe, you are saying what you see, but when it's linked to a graph you have to include data. So you might be describing between which set points on the graph there is an increase, a decrease, a plateau, a difference between means, for example. Explaining, you are saying why that pattern exists, and this is where you would then apply your biology knowledge to explain the pattern. Calculate, that's maths, and then conclude is you are saying what the experiment and the graph actually tells you. So we'll have a look at the first question, and this is a calculate question. So we're told that the glucose concentrations in different drinks has been investigated, and the results are shown in our graph. So we've got 5 different drinks, the concentration of glucose in grams per decimetre cubed. So the first question is, use the data from the graph to calculate how many grams of glucose would be in 150 centimetres cubed of drink F. So the first thing just to be aware of is they are giving us a volume in centimetres cubed. The volume that we have in the graph is decimetres cubed. So at some point you will need to do that conversion. So if I was doing this question, that's the first thing I'd be highlighting. Then it's looking at F in particular. So that's the one that we are looking at. So first step would be converting the units, as I said. So 150 centimetres cubed into decimetres cubed, we need to divide by 1,000. So we're actually working out how many grams of glucose are there in 0.15 decimetres cubed. Now the graph, if we read off, that tells us how many grams there are in 1 decimetre cubed, but we have 0.15. So that is why we would actually do 5 grams, because if we read across, ideally with a ruler, but don't have one on the computer, we do 5 grams multiplied by what we actually have, 0.15. So the answer is 0.75 grams. The next calculate question that we have is calculate how much more glucose is in drink D compared to drink E. So that's the first part. The second part is you then have to turn that into a percentage. So step 1, let's just find what the raw difference is. So D, if we have a look, that is 15 grams per decimetre cubed, whereas E, with your ruler, you'd read off, and we have 10 grams per decimetre cubed. So we have a difference of 5 grams per decimetre cubed. But we need to say what that is as a percentage. Now, because they said calculate how much more glucose is in drink D compared to drink E, that is why we're going to divide it by E, because the way they phrased it, it sounds like E is your starting point and we're comparing it to D. So it'd be 5 divided by 10, multiplied by 100 because it's a percentage, so it's 50%. Now that would be how you do the calculation to work out percentage change. Now you might have been able to do that in your head if you are quite mathematical, just by looking at the fact that this one contains 10, this one's 15, half of 10 is 5, we've got 5 extra, so it's a 50% increase. So either way you did it, the answer is 50%. So next then, we've got calculate and explain in this question. Scientists measured translocation in the phloem of trees by tracing using radioactively labelled carbon dioxide. So it's an isotope 14C. The volume of 14C but this is a carbon isotope that's incorporated into carbon dioxide. Release from the top and bottom of the main trunk of the tree was measured at regular intervals. And we can see that along here, the time after the labelled carbon was entered as carbon dioxide, and it says hours. So we've got hours as our time unit. So first of all, how long did it take the 14C, so that isotope, to get from the top of the trunk to the bottom of the trunk, and explain how you reached your answer? Now this question, what they're after is, you would need to go from the peak of your top trunk to the peak of the bottom trunk. The reason being is, and you don't actually have to explain this for your answer, but the reason I've gone from the peak to the peak is where we have this very, very high mean volume at the top of the trunk, we then need to look at where is the highest mean in the bottom of the trunk, because that will be when this set carbon actually made it to the bottom. And that's why I went from peak to peak. So that difference is 30 hours. So that is a calculate and then explain question. So it's a 2-in-1, that one. Scientists wanted to calculate the mean rate of movement down the trunk, but what piece of information are they missing? So the rate is always product over time. And we do have here the product over time. However, they've also stated it's the movement down the trunk. So that is the missing piece of information, the distance, which is the movement down the trunk. So it was the length of the trunk or the distance of the trunk. So overall, the units would be the volume of carbon dioxide, so that's our product, per distance, per unit of time, which in this case would be hours. So question 3, we're looking at the effect of temperature on the rate of an enzyme-controlled reaction, and that's what they've investigated. At each temperature, the reaction started using the same volume of substrate solution and the same volume of enzyme solution. So here are our results. We've got the concentration of product in the mixture in grams per decimeter cubed. And then we've got the time after the start of the reaction, and that is measured in seconds. So describe and explain the differences between the two curves. And this is a 5-mark question, but there is quite a lot of information to give here. So if we start with a description, this is where you say what you see. And this is describing the differences, remember, not just describing the two curves or lines. So the difference for 37 degrees C, we can see that there's a faster initial rate of reaction shown by this steep gradient. However, it plateaus after about 180 seconds. However, for the 25 degrees C, we can see the rate of reaction remains consistent the entire time, but it is a lower rate of reaction to begin with, shown by the fact that the gradient is less steep. So that would be our description, but then we have to explain why we have those differences. So there's our description. The reason then is, why do we have that faster initial rate of reaction? Well, it's at a higher temperature. Therefore, there's going to be more kinetic energy. So that means you'll have more successful collisions between the enzyme and the substrate, or you could phrase that as, you'd have more enzyme substrate complexes forming, which is the key phrase to always include in any explain question linked to enzymes. The second descriptive point as the difference was the graph plateaus at 37 degrees C, but it doesn't for the 25 degrees C. So we've got to explain why it plateaus, and that would be because we've got a different limiting factor now. And in this case, it is the substrate. So by that point, all of the substrate has already been used up and the reaction has already occurred. So that is our limiting factor. So those would be your 5 marks. Question 4, we've got another calculate. So you can see that you do get lots of calculate questions linked to graphs. The graph below shows the results of an investigation into the concentration of protein in blood plasma for people between the ages of 60 and 95. And the bars show 1 times the standard deviation above and below the mean. So we have males, females, we've got our standard deviation. Our axes are the mean concentration of protein in the blood plasma in grams per decimeter cubed, and we've got age in years. So we have to use the graph to say what is the difference between the rate of change of the mean concentration of protein in the blood plasma in males and females between the ages of 60 and 95. Show your working. So there's quite a bit there just to get your head around first of all. So number one, we are working out a difference, and it is the difference between the rate of change. Now the rate is not on the graph, so that's one thing we are going to have to calculate, the rate of change, and that is the rate of change of the mean concentration, which is what we have on our y-axis, of protein in the blood in males and females. So that's what we're comparing, males compared to females, and the time frame for our rates is 60 to 95. So step one, let's work out what the mean concentration rate is for females. So you need to work out the difference first of all before we then turn it into a rate. So the mean concentration was 71 to begin with in females, and then that went down to 60, so that's a difference of 11, but that was over 35 years. So to turn it into a rate, we divide by 35. So 0.31 grams per decimeters cubed per year is the rate for females. Do the same thing but for men. So we've got the difference in concentration is 69 minus 64. So it decreased by 5 grams per decimeter cubed over 35 years. So their rate is 0.14 grams per decimeter cubed per year. So that's the first bit, but they have actually asked for what is the difference between the rates. So the final step is working out what is the difference between 0.31 and 0.14, which is 0.17. And now at the final step, when you give your answer, you do have to write down the units, and that is grams per decimeter cubed per year. And one thing, when you're working out the units, just think about what did you do to your answer, and that's what you do to your units. And what I mean by that is we did the y-axis divided by the x-axis. So that's what we need to do for our units. Grams per decimeter cubed divided by years, which you could just do the division symbol and write year there. But year to the -1 means per year. So it means the same thing. Okay, question 5, and this is our first conclude question. Now it's the same information as the question before, so it's again looking at concentration of protein in blood plasma for people between the ages of 60 and 95, but this time you are going to have to conclude something. So a student concluded from the graph that the protein content of blood plasma decreased with age more in females. So that is their conclusion, and you've got to say whether you agree, and it's 2 marks. Now we haven't used the standard deviation yet, and if you are coming to a conclusion, you do need to use standard deviation. So that's the key thing that we're going to look at. So this student has said that the blood plasma protein content decreased more in females. Now we did see that there was a decrease of 11 in females, and there was only a decrease of, I think it was 5. We said it went from 69 to 64. So yes, that is a bigger decrease. However, the standard deviations is going to be key here. And with the standard deviation, if the bars overlap, what that means is there is not a significant difference between those means. Now it's normally represented as a bar chart, so this is actually quite different here. It's shown as a line graph. So although the mean here is different, we have for males it's at 69, whereas for females it's at 71, their standard deviations overlap, so that is not a significant difference. And that is the same at every single age except for 95. So that will be what the second mark is, that there's actually only a significant difference between the means at age 95. So although we have said that decreases more in males than females, decreases more in females than males, none of those decreases are actually different until age 95. So that is what the second mark would be. So final question is an explain one. The graph shows the oxyhemoglobin dissociation curves for two different species, a human, which is species A, and a rat, which is species B. Now species B, which is the rat, is more active than species A, the human. And you have to use the graph to explain how the hemoglobin of the rats allows a greater level of activity. So this is applying your knowledge of what you know about more active animals and how their hemoglobin would differ, and use evidence from the graph to support that. So the first piece of evidence would be pointing out that for species B, their oxyhemoglobin curve is to the right. So that's the first mark. The curve is further to the right, and what that tells you is the hemoglobin has a lower affinity for oxygen. And the way that we know that is if you just pick a partial pressure. So I'm going to go for 6. At the same partial pressure, which just so happens to be 6, we can see species B is only 30% saturated, whereas species A is around 50% saturated. So because species B has a lower saturation, it must have a lower attraction, a lower affinity for oxygen. So that is what the second mark is. Therefore, it means the hemoglobin must be unloading or dissociating the oxygen more readily. Now, the underline here more means you would have had to have said that to get the mark. The reason being is species A is also unloading oxygen because it's only 50% saturated, so it has unloaded half the oxygen from the hemoglobin. However, the difference is species B is unloading it more because it is less saturated. So the reason that's an advantage is more oxygen, and again you have to say more because oxygen is being delivered to the cells for humans as well. So more oxygen is delivered to the cells, or you could say tissues, muscles for the rats, the species B. And that then links back to what they've said in the question. The rat is more active. Explain how the hemoglobin allows it to be more active. Well, we've gone through points 1 to 3. The final thing is, because there's more oxygen available, that means they can respire faster and therefore have that greater activity. So that's one thing just to be aware of. If you do have a comparison, you have to have more, and that comparative word in there. The 15-mark comprehension at the back of paper 2 for AQA A-level biology is notoriously one of the hardest questions on the entire A-level. So in this video, I'm going to be talking you through my exact strategy of how I approach the comprehension questions so that you can do exactly the same thing in paper 2 and try and get as many of those 15 marks as possible. Hey everyone, and welcome to Miss Estruch Biology. And in this video, I'm going through everything comprehensions, my top tips, and how to get as many marks as possible on this. Now, if you do want even more advice on the comprehension, because let's face it, it's 15 marks of that paper, which is a big chunk, then let me tell you about the 1-hour lesson that you can actually watch, which is the recording of the Easter bootcamp that I did. In that 1 hour, I go through exactly how to answer the application question with a modelled example, and you get a workbook with modelled examples, the mark scheme, examiner's feedback to help explain the mistakes students made and how you can avoid those mistakes. And if you do want that, then I'll link it in the description below so you can get your hands on that full hour comprehension lesson. But for now, let's get into the key facts about the comprehension. Number 1, it is 15 marks. So there'll be a selection of questions linked to the comprehension. Collectively, they add up to 15 marks. It's on paper 2 and it is right at the back of paper 2. After the text, it always then says, use the information in the passage and your knowledge to answer the following questions. So essentially they are all application questions because you're having to use the text and your knowledge to be able to answer the questions. It's usually 15 to 20 lines worth of text, and they number every 5 lines in the paragraphs so that they can then direct you to which lines to look at specifically when you're answering each individual question. And then lastly, the questions tend to be application, but also I have seen maths questions come up linked to the comprehension as well. So those are the key facts. Next then, let me go through my key strategies for answering the comprehension questions before I do a modelled example for you. So the first thing I recommend is read through the entire block of information, but as you read through, highlight as you go. And the sorts of things I'd be highlighting are what you think might be a key term or a key hint at a topic that it might be linking to. So highlight the information as you're reading through it. And then linked to that, my second tip is annotate as you go. So some of the highlights, I might not have an annotation because it might be fairly obvious. I've highlighted enzyme just to point out to myself that this topic's going to be about enzymes, where it might be linking to transcriptional factor enzymes, for example. But annotations are when you might have a bit of information where I stop and pause and think, what is the relevance of that sentence? And I'll try and work out which topic it might link to, or if it's to do with an experiment, I might work out, is that the independent variable? Is that the dependent variable? Is it a control variable? Is it a controlled experiment? So I start to consider the information I've been given and try and work out what is the relevance of them giving me this fact, or what bit of information does that link to. Now that'll make more sense when I do the worked example with you very shortly. Tip number 3 is do not start on the comprehension. It is at the back of paper 2. It's worth 15 marks. It should take about 20 minutes, but I do not suggest you start on it, which that might sound obvious because it's at the back, but the reason I say that is my tip for paper 1, which has already happened now, is start at the back on the long answer questions. But I do not recommend that for the comprehension because it is one of the hardest questions on the paper and it's mainly application, so you are better off saving it to the end when your brain is already awake and active and warmed up. And also it means that you aren't going to maybe panic and spend loads of time on it and run out of time on the rest of the questions. So save it till the end. Ideally you should have 20 minutes left when you get onto the comprehension. And then my final tip is apply all the same strategies that I've talked to you about in my application videos, because these are basically all application questions. So if you haven't seen my application skills video, then I'll link it up here. Definitely watch that so you know the 4 key tips of what I recommend for how to approach all application questions to get full marks. So those are the key facts, those are the key strategies. Let's now go over to me answering some of these questions to model it for you so you can see how best to apply it and improve. So let's go through this example then. And as I said, I'm going to be going through it with the workbook that comes with the 1 hour lesson. I won't go through the whole thing because obviously that was a 1 hour lesson. So I'm going to skip past some of what we already talked about and get to the practice question. So let's go through this example together. I'm going to go through what I would highlight and annotate and why from the block of text, and then just go through this first question. But as I said earlier on, that if you are interested in having a go at more of these, then you can get the comprehension lesson and you've got the full pack of 3 example comprehensions with the mark scheme, common mistakes and explanations as to why that would be a mistake. But I'm just going to go through this first part with you, the information and question 1. So let me get the highlighter at the ready. North American black bears can hibernate for up to 7 months without needing food or water, relying on fat stored in their bodies from the summer. So the things I'd be thinking about here is if they're hibernating without needing food or water and they've got fat stores, yes, this is year 13 content, but I'd be linking it maybe to somehow to respiration. I'd be thinking, so if I was going to annotate onto this one, I'd be saying food and water, glucose is required for respiration, fat stores. So if you run out of glucose, then you can hydrolyze the triglycerides into glycerol, which can be used in the Krebs cycle. So if I was going to annotate something, I'd just write respiration, glycerol as a respiratory substrate. Then we've got during hibernation, their heart rate drops from an average of 55 beats per minute in the summer to 14 beats per minute and their metabolic rate decreases by 75%. So again, if we're thinking about metabolic rates, I just write again respiration here, and linking to the 55 beats per minute, I might just make a note to myself, control of heart rate, and thinking about the fact that the heart rate is going to drop, linked to the fact that there's lower respiratory rates, meaning you need less glucose and oxygen being transported. Next, then we have uncoupling proteins. Now, this is an unfamiliar term, so I'm just going to put that in highlights so it stand out to me if I needed to come back to it. And we're told they're present in many mammals, and they help mammals to maintain a stable body temperature during hibernation. So I'd highlight here and annotate homeostasis. These proteins are found in the inner mitochondrial membrane. So I'd highlight that and write oxidative phosphorylation, because that's the stage that happens there. In fact, it happens on the electron transport chain, is those proteins embedded within the membrane. So I'd annotate that. And it acts as a proton channel during chemiosmosis, but they do not produce ATP. So what I'd be thinking here is the proton channels, that's where the energy released from those electrons moving along pump the protons to the other side of the membrane in chemiosmosis. But if we're told that it doesn't end up in the production of ATP, I'd probably write a little note to myself here, just question mark, what happens to the energy then? If the energy is not being used to make ATP, what is it being used for? Then the last paragraph, in the mountains of North America, snowshoe hares change their fur colour from white to brown as winter turns to spring. So if they're changing from white to brown as winter turns to spring, the sorts of things I'd annotate here would be adaptation for survival, maybe some concepts of natural selection, maybe something linked to inheritance as well. However, earlier snowmelt due to climate changes has reduced their survival rate. So yeah, again, I definitely link this to natural selection, which yes, that is a paper 1 topic, but speciation is paper 2. So it might be more linked to speciation. This colour change, known as molting, occurs when new fur grows in. Recent research shows that within a population, snowshoe hares molt at different times, which may be key to their survival. So if they're molt at different times, key to their survival, yeah, I think that's adaptation, natural selection, maybe speciation. Use the information in the passage, so you always have that sentence. So suggest and explain how black bears are able to hibernate for up to 7 months without food or water, which is lines 1 and 2, and it's a 3-mark question. So we then look back at what we've highlighted, and I was saying, thinking about linking it to respiration. Those fat stores could be hydrolyzed. The triglycerides are hydrolyzed into glycerol, and the fatty acids and the glycerol could be used in respiration. So that's what I'd be using as part of my answer for this. But let's have a look at, you can always pause and have a go at this yourself. I'm going to go straight on to what the mark scheme is and look at the common mistakes and explanations behind it. So first of all, there was one link to the concept of respiration that we said. So the fat is used in respiration. Then we've got less energy or food storage required due to a low respiratory rate. So we talked about that at the beginning when we said the annotations would add. Then you've got the gluconeogenesis. So that's the description that I was giving, that the glycerol from the lipids could be used. Then other concepts we've got. So I actually didn't address the water, which is what this bit here is talking about. Maybe they've got a long loop of Henle, so less water is lost. Water is provided as a product of respiration. Maybe they don't urinate as much. They're not going to have as much evaporation. So that's one bit that I didn't actually talk about, was addressing the water side of things. But then we've also got the low surface area to volume ratio there. So common mistakes. Students attempted to describe gluconeogenesis, but very few did this successfully. Most confused this process with glycogenolysis. So just an explanation here that gluconeogenesis is the creation of glucose from a non-carbohydrate molecule. Glycogenolysis is the hydrolysis of glycogen into glucose. And the reason this question it had to link to this was, in the information they told us, they rely on fat stores, not glycogen stores. So that's how you knew it had to be gluconeogenesis, not glycogenolysis. Then we have several features of hibernating bears were mentioned, including reduced movement, fat insulation and their reduced surface area to volume ratio, but the significance of these features were rarely explained. So the question said to state and explain, so you had to give the reason why and not just the feature. And in this video, what we're going to be focusing on practicing is the extended response questions and a little bit on the long answer questions as well. So if you are new here, click subscribe so you don't miss out on any of the future exam technique videos. So here we see a glance at the specification, just so you can see how this links to AQA A-level. However, if you're doing another exam board or a different qualification, you will still probably come across long answer and extended response questions, so this will help you. But what I'm going to be focusing on is how to approach the extended response questions. And if you have a look for paper 1, 2 and 3, they do all include a mixture of short and long answer questions. For paper 3, it's more critical analysis though. And the long answer questions can be based on any skill really. It could be testing your knowledge, getting you to apply your knowledge, or even critical analysis. The extended response questions I'm going to show you later on in the video, but they're mainly assessing your knowledge. And that links to one of the top tips that I'm going to be going through. So it's 15 marks on paper 1, and let's have a look at some of these. So I've got here lots of examples of these extended response questions which have been on past papers for AQA. And on the slide before, you could see that it said 15 marks of that paper will be extended response. And that will either be a combination of 3 5-mark questions, or a 4-mark, 6 marks and a 5-mark question. And having a look, you can see, as I said, they're mainly knowledge based, so they're just testing what you know. And we can see from these command words, we've got define, describe, explain, compare, contrast. They are the lower level skills in the exam technique. And just to clarify exactly, I've gone to this website, which is the AQA website, and I'll link that in the description so you can go there as well, just to refresh your knowledge on what those command words are actually asking you to do. So define is specify meaning, describe, give an account of, explain is give reasons, compare could be looking at similarities or differences, contrast is just identifying the differences. So as I said, these questions at the back of the test, the extended response, are really just assessing your knowledge. So for that reason, my first top tip out of the 5 is paper 1, start at the back. Go straight to those 15 marks of extended response questions, because assuming you have revised a lot, you should have the knowledge to be able to get nearly full marks or full marks on those questions. And paper 1, students often find there's quite a tough time demand with that paper, because they will be spending extra time on other questions, reading through information, analysing graphs, applying their knowledge. Whereas if you go straight to the back, you don't then run the risk of running out of time and missing 15 marks that potentially you could have quite reasonably achieved. So the next one, bullet point or draw a table. I'm going to demonstrate this, but what I mean by that is you don't have to write continuous prose and long paragraphs in any question on an AQA paper except for the essay. So make it easier for yourself and the examiner so they don't mismark your paper by bullet pointing your answer or even drawing a table. Linked to this idea of helping you to make sure you've not missed any points, but also to make it as easy as possible for an examiner to mark your paper so they don't mismark it. Highlight what you think is a key word or marking point in each of your sections of the table or in each bullet point. And when I say highlight, you're not actually allowed to use highlighters in an exam. So instead it could be you write in capital letters, or maybe you just underline it. Top tip number 4, if it is a 5-mark question, put 6 points down. So you've got 1 extra as an insurance in case one of your previous 5 wasn't actually on the mark scheme. And finally, this is about your continual preparation. make sure you complete all of the extended response questions that are available on your exam board's website for both the AS and the A-level papers. Now that's going to help for a couple of reasons. Number one, you'll get much quicker at this idea of bullet pointing or drawing the table, highlighting the key word. So you'll become more time efficient before you get into the exam. Number two, it's just good revision of the knowledge. And then number three is really important. By completing all of those extended response questions, you'll get to the point where you can spot patterns in the mark scheme. And as I've been teaching A-level now for over 10 years, I'm well into that stage where I can spot patterns, and if I see a particular question, I already know what the mark scheme is going to be, just from experience over and over again of seeing different versions of that question. So for example, when it comes to any question to do with enzymes, what I always tell my students is have one of your marks linked to the idea of enzyme substrate complexes. Now I've just written ES there for short, but you would have to write enzyme substrate complexes, because that is one key word or key phrase that is always on the mark scheme. Similarly, with any question linked to proteins and explaining the function of proteins or describing the structure, there are always marks for correctly describing the bonds in each level of organisation, so primary structure, secondary structure, and so on. And you too will start to notice those patterns with practice. So let's have a look at a worked example. And I've picked out this question. Contrast how an optical microscope and a transmission electron microscope work, and contrast the limitations of their use when studying cells. And this was actually a 6-mark question. So one way you could do it, I said, was bullet points. So on that page where you have the question, I would split it straight away into these 2 headings to make sure that I was addressing both parts of the question. So first thing then is we'll have a look at how they work. And because you've been asked to contrast, we have to point out differences. And whenever you are contrasting or comparing, you need to have the comparative or contrasting points all in one sentence, and therefore there should always be whereas in the middle. If you only talk about one side, so for example, if you just talk about the optical microscope and don't talk about the electron microscope, you won't get the full marks. So how they work, I've got a beam of light is condensed to create the image for an optical microscope. Now I need to contrast, whereas a beam of electrons is condensed to create an image for an electron microscope. The next thing, a beam of light in an optical microscope is focused using a glass lens, whereas it's electromagnets for the electron microscope. So that's straight away 2 points, and it addresses the first part of the question. The second part is about limitations. So here I have a selection of limitations. And again, because it's a contrast question, in each of these bullet point sentences, there has to be that contrasting joining words. We've got here whereas, lower, longer, shorter, so you are showing it's a comparison. So I've got an optical microscope has a poorer resolution, whereas the electron microscope has a higher resolution. This is due to a longer wavelength in the optical microscope because of light, and a shorter wavelength because the electrons have a shorter wavelength in the electron microscope. Now, although that's one bullet point, that's actually 2 different ideas. So that would be 2 points. So we're already up to 4 bullet points to address a 6-mark question. We then go on to 5, 6, 7. So again, I've done 7 points instead of 6. So I've got that insurance, that backup. And then I've got the idea of lower magnification, colour image compared to black and white, living samples compared to dead samples. Now, when I said on the top tips, if I just go back, bullet point, you've already seen that. I've shown you that I've done 1 extra mark, so 7 marks instead of 6. Now, the highlighting, the key word, that's what I've done here in bold. And if it was written, I'd be underlining it. So I've underlined what is the concept or the key word or the feature that I'm referring to. And I would actually in the exam as well write in capital letters where you have that comparison phrase. So we've got longer, shorter, lower, whereas, to really emphasise you are contrasting. Now, the alternative is, if you don't like the bullet points, do it as a table. You do not get penalised. You are absolutely allowed to write your answer as a structured table. It makes it so much easier for an examiner to mark it, that they are less likely to miss something you've written. You're less likely to miss a concept or an idea that you want to add for your answer. And the way I've done this one, you don't have to write whereas, because we've already got the comparison. So for example, a beam of light is used to create the image, whereas for the electron microscope, here is a beam of electrons. So you don't need to have the whereas, because we are quite clearly showing the contrasting features for the optical compared to the electron microscope. Same idea with all of those limitations I've said. I've got the comparative or contrasting point directly next to each other on the same row. So I'm indicating that those are the contrasting features. So see what works best for you. And for some questions, a table might not work, it might not be appropriate, and it might be bullet points are better. But absolutely go for bullet points or a table to save yourself time, to help make sure that you are including enough marks, and to indicate the key words and to make it as easy as possible for an examiner to mark your paper.